Welcome to the Biotechnology lecture series. This is the second lecture on cell biology. In this lecture, we will cover meiosis and mitosis, their introduction, cell division, characteristics, and stages. And at the end, we will discuss some quiz questions and vocab. Have you ever wondered how living organisms grow, repair themselves, or reproduce? The answer lies in the process of cell division. Simply put, cell division is the process by which a cell divides into two or more daughter cells. All living things, from the tiniest bacteria to the tallest redwood tree, depend on this fundamental mechanism for growth and development. So, let's dive in and explore the fascinating world of cell division. Cell division is the process by which a single cell divides into two or more daughter cells, each with the potential to grow, develop, and carry out specific functions. Cell division can occur through two main mechanisms: mitosis, which produces two identical daughter cells, and meiosis, which produces four genetically diverse daughter cells. It is a fundamental process in biology that is essential for the growth, repair, and reproduction of living organisms. Mitosis is the process by which a single cell divides into two identical daughter cells. This type of division is essential for growth and repair in multicellular organisms, as well as for asexual reproduction in some single-celled organisms. And meiosis is the process by which a single cell divides into four daughter cells, each with half the number of chromosomes of the parent cell. Meiosis is critical for sexual reproduction, as it allows for the production of gametes. There are several characteristics of mitosis. Number 1. Mitosis produces two genetically identical daughter cells from a single parent cell. Number 2. The major purpose of mitosis is for growth and to replace worn-out cells. Number 3. Mitosis occurs only in somatic cells, which are all the cells in the body, except for the reproductive cells. And number 4. There is no reduction in the number of chromosomes during mitosis. Mitosis plays a crucial role in the growth and development of multicellular organisms, enabling cell division and proliferation, ultimately contributing to the formation of tissues, organs, and organ systems. In instances of tissue damage, like cuts or injuries, mitosis facilitates repair and maintenance by replacing damaged cells with new healthy ones. Furthermore, mitosis is instrumental in asexual reproduction for certain organisms, ensuring the production of genetically identical daughter cells and maintaining the offspring's genetic similarity to the parent cell. There are almost 5 stages in mitosis. The first stage is prophase. During prophase, the chromatin in the nucleus condenses into visible chromosomes, which become visible under a microscope. The centrosomes, which are responsible for organizing the spindle fibers, move to opposite poles of the cell. The nuclear envelope breaks down, allowing the spindle fibers to attach to the chromosomes. During metaphase, the chromosomes line up along the equator of the cell, called the metaphase plate. The spindle fibers, which are attached to the centromeres of each chromosome, ensure that the chromosomes are properly aligned and can be separated correctly. During this stage, the sister chromatids of each replicated chromosome separate and are pulled towards opposite poles of the cell by the spindle fibers. This results in the formation of two identical sets of chromosomes at opposite poles of the cell. During telophase, the chromosomes reach the opposite poles of the cell, and the nuclear envelope begins to reform around each set of chromosomes. The chromosomes begin to uncoil and return to their normal diffuse form. The spindle fibers disassemble, and the cell begins to prepare for cytokinesis. Cytokinesis is the process by which the cell divides into two daughter cells. In plant cells, a structure called the cell plate forms along the equator of the cell, eventually dividing the cell into two separate daughter cells. In animal cells, a contractile ring of actin and myosin filaments forms around the cell, contracting and pinching the cell membrane until the cell is divided into two separate cells. Here are some vocab that you should remember. Like cell, it's the basic unit of life and the smallest functional and structural unit of living organisms. Split chromosome is a chromosome that has undergone a structural change, resulting in the separation of the chromosome. A chromosome is a structure found in the nucleus of cells that carries genetic information in the form of DNA. A centrosome is a small organelle found in eukaryotic cells, which functions as a microtubule organizing center. The spindle fibers are microtubules that separates the chromosomes into two daughter cells. Centrioles are cylindrical organelles found in eukaryotic cells that are involved in the organization of cell division. Here are some quiz questions. Read each question carefully and select the best answer. What is mitosis? 1. The process by which cells divide into two identical daughter cells. 2. The process by which cells differentiate into different types of cells. 3. The process by which cells die and are replaced by new cells. Second question, what are the 4 stages of mitosis? Gon, Si, G2, M. 2. Prophase, metaphase, anaphase, telophase. 3. Cytokinesis, interphase, csir, M. Third question, during which stage of mitosis do the chromosomes condense and become visible? 1. Prophase. 2. Metaphase. 3. Anaphase. Fourth question, during which stage of mitosis do the chromosomes line up at the center of the cell? 1. Prophase. 2. Metaphase. 3. Anaphase. To know the answers of these questions, search comment section. Mitosis is not just a process of cell division. It's a beautiful dance of chromosomes that ensures the continuity of life. That's all from this lecture. See you in the next video for lecture number 3. And don't forget to subscribe to this channel and press the notification button so you won't miss upcoming lectures.